In this video, we will discuss about the CD16-NK92. So in this paper, they are using two different kind of CD16 expression on NK92. First is cDNA of CD16 that is expressed on NK92, and second one is CD16 fused with CAR that is expressed on NK92. And we have discussed the results that are presented in this paper before this figure, that where they confirmed that the exogenous expression of CD16 through both the methods are bona fide or exist in the NK92 cells. So, and they use these lentivirus transductions to express those exogenous CD16 on the NK92. And once confirming the CD16 expressions, now they wanted to check whether this CD16 NK92 cells has cytotoxicity activity against the target cells or not. So in order to do that, they selected different kind of target cells such as K562, A549, Raji cells, Daudi cells, BT474, SKBR3, SKOV3, and MD458, which is a kind of breast cancers. These last two are the kind of breast cancer. And then they check this CD16 NK92 and CD16 NK92 cytotoxicity activity against the different target cells at different effector to target ratios. And those effector to target ratios are shown on the x-axis, and they have used all the three conditions, that is wild type NK92, CD16-NK92, and this is CD16-NK92 with CAR. And what they are able to see that, and in this experiment, they have not used any antibody to trigger antibody dependent cellular cytotoxicity. So what it is, they co-culture this NK92 and the different target cells at different effector to target ratio, and then check the cytotoxicity effect by means of this measuring the percentage lyse cells in the percentage lyse target cells in the co-culture medium. And what they are able to see, as we can see that in all the cases they are able to see almost similar results. Similar results means when they compare wild type NK92, CD16-NK92 and CD16-NK92 with CAR, that all the three conditions seems to be at the same point at their respective effector to target ratio. So this in terms indicates that this NK92 CD16-NK92 does not have enough cytotoxicity activity to trigger enhanced target cells death. So in order to do that, the idea that comes into the researchers is that if they can use some antibody to enhance the cytotoxicity activity of the CD16-NK92 cells, thereby they can be able to see and increase its cytotoxicity activities against the target cells when it is compared with the wild type NK92 cells. And one of the approach that they used in the next figure that we will discuss in our next video, that where they use the antibodies is Rituximab and one more drug. So what they did is basically these drugs will coat the for example, this Rituximab that we have discussed in the previous video, it has the CD20 plus expressions and it also has the Fcregion. So the CD20 plus affinities, CD20 plus receptors, we can say that it can bind CD20 ligands. It can bind to the target cells that has the CD20 expression on their cell surface, and these CD20 expressions will be detected by the antibody, and they will basically coat the CD20 positive target cells, And since this antibody, that is Rituximab, also had these Fc receptors, which is detected by the immune cells or the NK92 cells. So this will enhance the cytotoxicity effect by making target cells more recognizable to the immune cells. So this is what they have done in their next experiment, and those results we will discuss in our next video. So I hope you understood here how they check the cytotoxicity activity of CD-16 and NK92 and compare it with the wild type. However, they did not find any significant changes, even though they tested for like 8 different target cells. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.